meiosis, and they'll be in various kind of packagings on the top of an aerial hypha. So they're very similar. You just have these three extra steps in the process. What's that? Where is it? Oh, we'll see. We're going to see a visual example in just a moment. But this is this goes. If you're wondering, this is actually the product of this process here. So yeah, I should probably update the notes to throw that in there. But plasmogamy results in a dikaryote cell with 2 nuclei, which then fuse into one diploid nucleus and carry on. Okay, so now we're going to just look at some characteristics of 3 major fungal phyla. Again, this is another kingdom that's got a lot of different classifications depending on what resource you're looking at. So these are just 3 major ones. And there'll be others as well. Now, let's just put them up here and we'll look at each one briefly. We're going to focus on the first and the last one. Okay? This one we'll just briefly touch on, but it includes very familiar kinds of fungi such as the mushrooms. So the Zygomycota, that's the name of the phylum. They're commonly called the zygomycetes. They're, or more unofficially, the conjugation fungi, even though they're not the only fungi that engage in conjugation. So we'll look at those first. Then we'll just touch on the Basidiomycota. They're called the club fungi because they have little microscopic club-shaped structures where the spores are produced, as we'll see. And then lastly, the Ascomycota. Okay, and that they have a special, when they reproduce sexually, they have a special little sac. It's not a sporangium. It's a little different. It's called an ascus. So they're called the Ascomycota. And what does mycota, MYC, refer to? Fungi. Okay. So again, a few examples of the kinds of fungi you find in this group. So let's take a look at first the Zygomycota. And this diagram is from your book. And on the left, this is the cycle, the asexual reproductive cycle, and on the right, the sexual reproductive cycle. Okay, so again, at a glance, it looks complicated. It's really not. Okay, so here's one, and by the way, I think I have it up here, don't I? Yes. So characteristic of the Zygomycota or zygomycetes, they have aseptate hyphae, also known as cenocytic. Remember those terms? And they produce sporangia. Okay, so sporangia spores in a sporangium suspended at the end of a sporangiophore. Here's your sporangiophore, here's your sporangium, and all the sporangiospores or sporangia inside. So a single spore lands somewhere, grows by mitosis. Again, that's haploid, 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 haploid. Okay, and you get your mycelium and so forth, and then aerial hypha protrude upwards and develop a sporangium with sporangiospores inside, which ruptures and releases more spores and just repeats the cycle. Okay? So very straightforward. That's the asexual reproductive cycle of a zygomycete. Over here, if it happens to be near a mating partner, so they have sort of chemical means to distinguish each other. They're kind of like boy and girl moles. You have plus and minus. Sometimes there's alpha, beta, but whatever they're called, they're opposite mating types. And so here we go. So they recognize each other, and guess what's going to happen here? Okay, you got a haploid cell, haploid nuclei. What's going to happen? What just happened from here to here? Plasmogamy, resulting in that. Okay, a dikaryon. And then, from here to here, we went from 2 haploid nuclei, one from each partner, in a dikaryon to a single fused diploid nucleus, a result of karyogamy, nuclear fusion, as opposed to cytoplasmic fusion. Okay? And what happens next, you're going to have, as I was showing you about meiosis, and then a lot of mitotic cell division. You get what's called a zygosporangium. It's a very, you'll see these in lab in a couple of weeks. A very large, prominent, structure that's kind of brownish in color and looks has a rough-looking surface, almost looks like a little pine cone. And from that is going to sprout one of these, right? just like that. Okay? So you have sporangia 4 with a sporangium at the tip full of sporangiospores. And then that'll rupture and release and so forth. And then these can land and grow into a new fungal thallus that can either reproduce asexually or sexually. So that's the sexual reproduction in the zygomycota. We're going to see it's just slightly different when we look at the ascomycota in just a moment. So any questions so far? Actually, it's a misspelling. There should be an I in there. Basidiomycota. Again, mushrooms are a typical member of this phylum. What a mushroom is basically a structure produced as a result of sexual reproduction. So mating types meet underground. So they have extensive, mycelium or thallus underground, and then they reproduce sexually in the same basic way as we just saw. A bunch of aerial hyphae will, merge together to form the stalk and the cap and and so forth. And then in the gills underneath, microscopically, you have these little club-shaped structures with little spores at the end. So this is called a basidium, this club-shaped structure. So that's where the name comes from, basidia plural. And the spores will develop, again, they're haploid, a consequence of meiosis. They'll develop and then detach and can spread and grow into another new mushroom. So really nothing terribly new here except the nature of the fruiting body, as it's called, and these little structures here, the club-shaped fungi. So that's why they're called club fungi, not because this looks like a club, but because these little things look like clubs under the microscope. And the ascomycota. So just to kind of reinforce the whole process again, you have on the left asexual, on the right sexual. And on the left, okay, you've got a little spore, and it lands and it grows by mitosis, and it's gonna, got vegetative hyphae, and then it's gonna have aerial hyphae that are produced going upwards. And here's your conidia 4, and at the tips you have these little conidia spores here at the tips. And there's no, or conidia for short, there's no sac. So asexual reproduction looks different in the ascomycota compared to sexual, as we'll see in a moment. Whereas in the zygomycota, notice they're both going to be in a sporangium, okay? Sexual or asexual. So, and then these released and so forth, and it can grow into a new one. Sexual reproduction, again, involves opposite mating types, so you have your plus and minus up there, and maybe it's hard to see, but you've got right there, you've got what? You've got a two nucleus cell, a dikaryon. Okay, so plasmogamy has occurred, cytoplasmic fusion. And in this case, it's a little bit different in that, what do you see here? The dikaryon is reproducing by mitosis. So karyogamy has been delayed a little bit. So it doesn't have to happen immediately. Karyogamy can be delayed, a little mitosis in between. Eventually, though, at the ends here, the nuclei will fuse. Then you have karyogamy. So these are still dikaryons here, cells with two haploid nuclei. But at the tips, you get nuclear fusion, karyogamy. You get the diploid nuclei, and then they're going to undergo meiosis, and you get little haploid spores in a smaller sac called an ascus. Okay, so you don't have an ascus over here, but you do over here. Okay, and sometimes, it depends on the species, sometimes the 4 haploid spores then reproduce by mitosis, and you get 8. So some species there's 4 spores in an ascus, some there's 8. And then, they're eventually it ruptures and releases, and the spores can land and grow into a new fungal thallus. So again, it's the same basic thing: plasmogamy, a little more delayed, karyogamy, meiosis to give you some haploid spores that have a new genetic makeup, a little bit from each partner. Okay, and what else to mention about the ascomycota? I think it's up here, isn't it? Yes, their hyphae are septate. Okay, so they have very clear septa between each cell, or dicharion. When they reproduce asexually, that's where you're going to see cnidiospores. You don't see cnidiospores when they reproduce sexually. When they reproduce sexually, they're in an ascus. Okay? And all of the yeasts, what's a yeast? In 3 words, what's a yeast? Single-celled fungus. So they're all in this group here. All right, so hopefully that illustrates, the nature of sexual reproduction in a mold. And this is called, sexual reproduction is referred to as conjugation. So here's yet another conjugation. I'd like to say, just for the record, the next slide is on a slightly different type of yeast. Just skip that slide altogether. We'll just focus on this slide. So, there are different yeasts. There are a couple in particular that are very relevant to, humanity in various ways. In particular, the budding yeasts. Actually, I'll say one thing about the budding yeasts. Again, these are different species we're going to look at. The budding yeasts, when they reproduce, here's the cell cycle. Again, don't worry about the details here. It's just kind of showing when they reproduce, they form a little bud in the cytoplasm that gradually grows, and then as the nucleus divides, one nucleus will end up in the bud, and then the cells will undergo cytokinesis. You got 2 cells. This is sexual reproduction, or is it? Let me see. I'm sorry. They do reproduce sexually. They can reproduce asexually. So when they reproduce sexually, you know what happens? Opposite single-celled yeast meet, fuse, plasmogamy, nuclei fuse, karyogamy, meiosis, and then you get the 4 haploid cells as a result. So, that's to contrast them with what are called the fission yeasts. Okay, so when they divide sexually or asexually, they sort of lengthen, and then form. Cytokinesis will then occur in the middle like that. So they're called fissions, literally splitting in half yeasts as opposed to budding yeasts. Okay, so that's the distinction there. The budding yeasts, an example of a budding yeast is what we commonly call baker's or brewer's yeast. And here's its name. So you're gonna get a lot of names like this in the course, as you've seen. So good practice. Practice saying them. It's mostly Latin. A little Greek, that's been Latinized or something like that. So, Saccharomyces cerevisiae. You know what that fancy multisyllabic pair of words means? Sugar, fungus, beer, right? Cerveza. So it's brewer's yeast, and also used in baking, or baker's yeast. That's Saccharomyces cerevisiae. So I think we all can appreciate its value in baking as well as in creating fermented beverages. So it certainly benefits us in those ways. But I'd like to emphasize a couple of other things, a couple other ways in which our little friends, the sugar, fungus, beer yeasts, really do us, good things. Well, actually, yeah, for the record, they're facultative anaerobes. Okay? So what does that mean? They're capable and actually would grow a lot more efficiently if they have oxygen and can grow aerobically. But if there's no oxygen, if they're locked in a fermentation tank without air, oxygen, and just something to eat, they'll ferment. And their fermentation product gets us buzzed. That's why we do that to them. So we can drink their fermentation waste and have a good time, and hopefully not get sick and overdo it. But down here, I really want to emphasize a couple of things. Just for basic research, okay? So basic research. The way research works is you study something that's much to study, and then see how that applies to more complex organisms that might be more relevant to the general interest, okay, like humans, okay? So not a whole lot of research is done on humans, right? They're really a pain because they don't do what you tell them to. They don't like being locked up. They don't like being abused. Okay? They don't like being taken away from their families and had experiments done on them. So you don't do experiments on humans, okay? Even though, and plus they take too long to grow. It's like 20 years before you get babies and so forth. So it's a lot easier to study organisms like mice or chickens or fruit flies that reproduce very fast, and then ethically we can kind of do whatever we want. You don't want to be too harsh on the mice, for example. There are rules about that. But flies is like, whatever. Let the flies, flies, they reproduce very rapidly, and you can do a lot of studies. In fact, research an organism that doesn't take a lot of money to cultivate or grow, and that have a very fast, generation time. You can get a lot done in a short period of time for not a lot of money. So that's why fruit flies and little microscopic worms and things like that are topics of investigation, because this is very practical. And biology really is biology no matter what the organism. Okay? When you get to multicellular organisms, they all look very different and do different things, but when you get down to it, they're all made of cells that do the same stuff. So you can learn a lot about the way, for example, eukaryotic cells work by studying yeasts. Okay? A lot easier to study yeasts than humans or horses or whatever. Okay? Doesn't take much to grow them. Just get some basic nutrient material, dissolve it, put it in a flask, let them grow, and then you'll get billions and billions of them overnight, and you can do whatever you want with them. And we've learned so much about the way eukaryotic cells work by studying yeast. And just one aspect in particular, the cell cycle. Here's an example. You've heard of the cell cycle before, right? G1S, G2M. This is incredibly relevant to humans, because our cells undergo the cell cycle too. They divide and go through mitosis and so forth. But they're only supposed to do it when they're supposed to. And when they do it when they're not supposed to, you know what you have? You have a tumor. And when tumor cells acquire extra characteristics and can leave the tumor and deposit elsewhere in the body, you have a malignant tumor, or something we call cancer that, I think it's currently the #1 killer, or is it heart disease? I can't remember, but.
It's heart disease.
Okay, but they're right up there. A lot of people die from cancer. Everybody knows someone who's died from cancer or who's had to struggle with cancer. So we've made a lot of progress in dealing with cancer, and it all starts with understanding how the cell cycle works, which has been worked out very, very well in yeast. And then by looking in human cells, found out that, the same kind of proteins are doing the same kind of things in human cells. Now we have some targets we can, go after to see if we can, try to fix things in a sense, or get rid of cancerous cells. So anyways, just for basic biology, in particular the cell cycle, yeast have been incredibly valuable. And then studying secretory pathway, all that stuff. All that we know because of organisms like these little yeast. Yes?
Why do cancers still form?
Oh, good question. That's a topic I can get carried away with. I'll try to be brief. I'm glad you asked that, because I really should address that. It's not really going to be a topic of this class, but basically there's a lot of genes that encode various proteins that have a role in regulating the cell cycle. And so something has to go wrong, either in the way those proteins function or the balance of how many there are. Okay? And there's a couple ways that can happen. Generally, it's the traditional way to think of it is mutations. Okay? So you need mutations in different kinds of genes in a given cell for it to be able to divide uncontrollably. And there are things you may have learned about, maybe not, but cells have a suicide program. It's got to be disabled somehow. Okay? Cells send signals and receive signals that tell them to grow, and some cells, their grow signal is stuck on, for example. There's all these various little, as they call them, checkpoints. So there's proteins that function like little policemen at each stage of the cell cycle, and they're supposed to hold the process until everything's verified to be okay. Because if you continue when DNA is not done being copied, or chromosomes are broken, or whatever, you're going to get unequal distribution of materials, of genes in cells, and things can go wrong from there. So those little molecular policemen they got to be screwed up, okay, and not be able to do their job. So there's a lot of things that go into it. And then there's issues like angiogenesis. You know, when you get a tumor, it has to develop its own blood supply. Then there's genes involved in metastasis, so that cells can detach and through the lymphatic system go to a lymph node and other places and seed tumors there and so forth. So there's a lot going on, but generally it comes down to mutations. What's a mutation? It's just some DNA sequence, some DNA in the gene is changed for some reason. Sometimes you inherit mutations, right? So some families there's a propensity for cancer to occur, right? There's a number of different kinds of genes that need to go awry to have a cancer occur in a cell. And some people are just born with a few of those. Not enough to start a cancer, but it's just a couple steps closer than other folks to getting to that point. But additional changes, additional mutations will have to occur along the way. So nobody's born with cancer. People may have a few extra mutations they inherit, so they need a few less than other folks to get to the point where that cell is dividing uncontrollably. Okay? So, and then another thing too is a lot of times, chromosome number can get, that's a whole other story. But it's not just changing nucleotide sequences, but it's chromosomes that break, having extra or missing chromosomes. Those are very common in cancers, and that really can screw up the balance of gene products being expressed, which can tip the scales, towards a cancer. Just to give you one last thought on that, to appreciate how it's not just a mutation in a gene that can contribute to a cancer. Think of Down syndrome. You've all heard of. It's got nothing to do with cancer, but it's an example of there's nothing genetically wrong in Down syndrome. You know what the problem is? It's an extra copy of a chromosome. So there's 3 copies of genes on that chromosome in that cell, in those cells, as opposed to 2. There's not necessarily anything wrong with those, but they're the wrong amounts and thus the wrong concentrations of gene products from those genes is really the problem with what's going on. So it's not so much the gene itself, but how many or how much of it's being produced. And that's another thing that can go wrong in cancer. So, okay, I did get a little carried away, but did that answer your question? It's an interesting topic. And then mutation, obviously you want to avoid mutations. They give you a lead apron when you go to the dentist and so forth to protect your gametes from radiation damage, which could add a few more mutations. But just for the record, mutations can happen anyways, thanks to just good old, plain old DNA replication whenever a cell divides. So we'll talk about that later. So you can't escape it, you can just try and minimize it. Okay, got all worked up there. All right, good question. Okay, lichens. So this will be our last, oh my gosh, I have to. Okay, so lichens. This is our last fungus we're going to look at. Lichens are very interesting. They're not that big a deal, directly to humans. But I did want to mention them for one reason, because ecologically they have a very interesting role. They have a very unique ability. We'll see in a moment. First of all, what are they? They're actually a symbiosis. Okay? Remember, parasitism, a parasite and host is also an example of symbiosis, but in that case, one benefits, one is harmed. Okay? This is a mutualistic symbiosis. Both benefit. Okay? So when you hear the word symbiosis, that's generally what you think of. Okay? Birds and bees and things like that. So you've got a fungus and an organism from a completely different kingdom. Okay? It can be photosynthetic bacteria or commonly photosynthetic algae that live within the phallus of this fungus, and both together in this symbiotic relationship is a lichen. So a lichen is really 2 distinct organisms that are mutually dependent on each other. We'll see an example of what they are, what that looks like on the next slide. And just don't worry about this. You've got lichens that have different kinds of appearances. I don't care if you remember if it's crustose or foliose. There's a whole bunch out there. But this is what I wanted to emphasize. Okay, they're important pioneers, which is an interesting term biologically. Every now and then, things happen out there in the world where a whole region is utterly destroyed. biologically. Everything there is good kill, gone. And all that's left is inorganic, rocky material. You know, for example, what's the most famous volcano? Well, there's two you probably could think of. Krakatoa? No.
Pompeii.
Oh, that's right, Pompeii. That's a movie. That's right. That's great. Pompeii or Mount St. Helens here in our country. Well, any volcano that blows, you know, everything within a radius of I don't know how many miles is just gone, and then you have nothing but ash and pumice left over. But we all know over time, life comes back. Things eventually start growing. You get forests and so forth. So, you know, I was Krakatoa, this Indonesian island, you know, that's blew its top and now it's coming back out of the oceans, and now it's an island full of life again. So all that rocky material, how did that happen? You have to have pioneers. So pioneers have to be able to get the whole process going to get life going, coming in. Something has to be able to grow on inorganic surfaces. You can't just plant trees in a volcanic wasteland. They won't grow, okay? Or much anything else. There are a few kinds of organisms that can grow in these environments, okay? And they're called pioneers. And the lichens are an example. Some mosses can do that too, to some degree. So why is that such a big deal? Because they start carrying out their photosynthetic activity and producing organic material that slowly builds up. And when there's enough organic material due to their activity, then the next sort of level of organism can start to grow. You might get little grasses or weeds or shrubbery or something like that. And after many, many more years, then you can get trees and so forth. So they start this process that's called succession, if you've ever heard of that. But it's a whole process of kind of replenishing a whole ecosystem from nothing. And it all depends on pioneers like these little guys. Okay? Like okay, so I think we're done with the fungi. Oh, that's right. And this, don't worry about it. Just lichen structure, I won't test you on. It's just showing all the hyphae here, okay, and a nice little protective environment in the middle here where the photosynthetic symbiont, as it's called, whether it's an algae or like cyanobacteria, is going to grow, and together they depend on each other. What's the advantage, by the way? Here's the fungus, and here's the photosynthetic, partner here. And in most symbioses, well, in this symbiosis, what's the fungus going to get out of this relationship? What's that?
Energy.
Right, energy that it couldn't get otherwise. Energy thanks to its photosynthetic partner, which can, carry out autotrophic, right, production of sugars and other things that can then be passed on to its partner. What's the benefit of this one? Is it a slave? Not entirely. In fact, it's well adapted to this internal environment. It's protected and can thrive in here, whereas by itself outside can't. So, in fact, another such relationship I'd like to mention that I'm not going to test you on, but just for your information. Corals. Coral reefs are based on corals. And corals are, what kind of organisms are corals? Coral, what kingdom? They're animals, actually. They're animals. Corals are animals. But they have their brilliant colors because within their tissues they have a mutualistic symbiosis with photosynthetic algae. And they give the pigmentation and so forth. And they both depend on the other, and they can't live without each other. And again, one's photosynthetic and provides carbohydrates, and the other one provides a safe environment for its little partner. So very much the same kind of thing. Okay, conidiospores. What are they, and what phylum might you find them? So a conidiospore. You have a conidiophore, and then you have, what's that? Right. Vic, right?
Yeah.
Right.
It's the wrong part there. Strength.
Right. So there's spores produced at the little tips. Yeah, they have no sac whatsoever. Exactly. And so there's spores produced without any kind of sac. And under what conditions are they produced? Two choices. Asexual, exactly. And so the other part of the question was, in what phylum do you see such spores produced asexually? Well, right, so you got it, the ascomycetes, or also called the ascomycetes. For example, penicillium, you've probably heard, that's the source of penicillin. Penicillium's a mold, it's in the phylum Ascomycota, and you're going to see some of their conidiaspores in lavender.
They don't have that sac they're just like hanging on time.
Right, so they're just going to be sort of produced and then just detached from the end, and there's no cover, there's no sac at all. Now, if such a mold was going to reproduce sexually, then you would find those spores in a little sac called a ascus. Exactly. Okay, the phylum which contains the yeasts. 2 important roles yeasts play from a human viewpoint. This is a good question because I'm going to clarify something for you. First of all, the yeasts are in what phylum? Same one, the Ascomycota. 2 important roles yeast play from a human viewpoint. I remember I've asked this question in the past on a quiz or an exam, and I'll get answers like beer and wine. So what I'm getting at is 2 distinct kinds of benefits you get, okay? So you can list off a lot of foods that have relevance to yeast, bread and beer, or beer and wine, or whatever. That's not to me, that's only one kind of thing, okay? Food production. Food or beverage production is one benefit. What other benefits? There's really 3 in particular I pointed out, okay? All right, studying basically just basic research into how cells work, how eukaryotic cells work, basic research. And I just realized I skipped over one. I do that sometimes. My apologies. Let me go back real quick. Okay, this is in the notes. And there we go. Okay, so I skipped that one. So recombinant DNA technology, you may have heard that term before. We have the capacity now to literally put genes, foreign genes, into an organism's genome. That's part of what we call recombinant DNA technology. So we can put genes to make human or other gene products in yeast. Insulin. Okay, insulin, where does insulin come from? The pancreas, right? And in the old days, wasn't a human pancreas. You had to get it from pigs, if I'm not mistaken. Yeah. So, but nowadays, now we can clone insulin genes, and get isolated and put them in the genome of yeast. So literally, there are yeast that express insulin, and then to produce insulin for insulin supplementation, you just grow the yeast that have been genetically modified to express the gene. And you can grow infinite amounts of yeast if you want, very easy. And then you just process them to purify the insulin. That's where it comes from. Okay, and insulin is not the only thing. There's many other things, hormones and things that you can get them to produce, and hugely important. Okay, so just for molecular genetic manipulation to produce very useful products, yeast are also valuable. So that was our 3rd use for yeast, or benefit from yeast, I should say, to answer this question. So food production, basic research, and then producing, medicinal products by recombinant DNA technology in yeast. All right, I think we're due for a break. Let's take a break. We do have one more little section here. I'll try to be brief when we come back about the worms, and then we'll start chapter 5. Okay. Oh, yeah. You want to see the questions? That's fine. No. Remember the pioneers. Yeah. And basically what alike it is. Okay? Yeah. Continuous spores? Yeah. So continuous spores, there's spores that are produced asexually, without any enclosed sac. That's the main thing in members of the Ascomycota. Yes.
I want to structure that. How do we have pioneers up there? How do they get nutrients to like?
So how do they what?
Like how do they get like vitamins?
Oh, the nutrients?
Like, yeah, lipases.
Well, I guess they get the fungal part attaches to the surface. And, you have CO2 and oxygen in the air. I guess there'll be